this video, we are going to learn about cancer, a different perspective of molecular biology related to cancer. So, here we could see a picture which represents nasopharyngeal carcinoma (NPC), and that's a malignant tumor arising from the mucosal epithelium of the nasopharynx, the uppermost part of the throat. NPCs can arise due to cigarette smoking or eating nitrosamine rich foods such as salt cured fish or result from an Epstein Barr virus or EBV infection. The section showing the NPC is stained with hemoxylin and eosin. So that's what is represented here. It's a type of cancer like nasopharyngeal carcinoma. So cancers cause about 1/5 of the deaths of the United States each year. Worldwide, between 100 and 350 of every 100,000 people die of cancer each year. Cancer results from failure of the mechanisms that usually control the growth and proliferation of cells during normal development and throughout adult life. Intricate genetic control system regulate the balance between cell birth and cell death in response to growth signals, growth inhibiting signals, and death signals. Cell birth and death rates determine the rate of growth and adult body size. In some adult tissues, cell proliferation usually continuously occurs as a result of constant tissue renewal strategy. Intestinal epithelial cells, for instance, live for just a few days before they die and are replaced. Certain white blood cells are replaced just as rapidly. The skin cells commonly survive for only 2 to 4 weeks before being shed. The cells in many adult tissues, however, normally do not proliferate except during healing process. Such stable cells like hepatocytes, heart muscle cells, neurons can remain functional for long periods or even for the entire lifetime of an organism. Cancer occurs within the mechanism that maintain normal proliferation rates malfunction to cause excess cell division. The losses of cellular regulation that give rise to most or all types of cancer result from genetic damage that is often caused by tumor promoting chemicals, hormones, and sometimes viruses. Mutations in three broad classes of genes have been implicated in the onset of cancer, like proto-oncogenes normally promote cell growth. Mutations change them into oncogenes, whose products are excessively active in growth promotion, and oncogenic mutations usually result either increased gene expression or production of a hyperactive gene product. Then tumor suppressor genes normally restrain growth, so mutations that inactivate them allow inappropriate cell division. A third class of genes often linked to cancer, called genome maintenance genes, are involved in maintaining the genome's integrity. So proto-oncogenes, oncogenes, tumor suppressor genes, and along with that, genome maintenance genes will be there, which are involved in the maintaining of process of genomic integrity. When these genes are inactivated, cells acquire additional genetic changes in an increased rate, and including mutations that cause the deregulation of cell growth, and proliferation, and lead to cancer. Many of the genes in these three classes encode proteins that help regulate cell proliferation, like entry into and progression through the cell cycle or cell death by apoptosis. Others encode proteins that participate in repairing programmed DNA. The cancer forming process called oncogenesis or tumorigenesis is an interplay between genetics and the environment. Most cancers arise after genes are altered by cancer causing chemicals known as carcinogens or by errors in their copying and repair. Even if the genetic damage occurs in only one somatic cell, division of this cell will transmit the damage to its daughter cells, giving rise to a clone of altered cells. Rarely, however, does mutation in a single gene lead to the onset of cancer. More typically, a series of mutations in multiple genes create a progressively more rapid proliferating cell type that escapes normal growth restraints, creating an opportunity for additional mutations. The cells also acquire other properties that give them an advantage, such as the ability to escape from normal epithelia and to stimulate the growth of vasculature to obtain oxygen. Eventually, the clone of cells grows into a tumor. In some cases, cells from the primary tumor migrate to new sites where they form secondary tumors, a process termed metastasis. And most cancer deaths are due to invasive, fast growing metastasized tumors. Time plays an important role in cancer. It may take many years for a cell to accumulate the multiple mutations that are required to form a tumor. So, most cancers develop later in life. The requirement for multiple mutations also lowers the frequency of cancer compared with what it would be if tumorigenesis were triggered by a single mutation. However, a large number of cells are in essence mutagenized and tested for altered growth during our lifetime. A powerful section in favor of this mutagenized cells, which in turn we in this case we do not want. Cells that proliferate quickly become more abundant, undergo further genetic changes, and can become progressively more dangerous. Furthermore, cancer occurs most frequently after the age of reproduction and therefore plays little role in reproductive success. So, cancer is common in past, reflecting an increasingly longer human lifespan, but also reflecting the lack of selective pressure against that disease. Clinically, cancers are often classified by their embryonic tissue of origin. Malignant tumors are classified as carcinomas if they derive from epithelia such as endoderm, gut epithelium, or ectoderm, skin and neural epithelia, and sarcomas if they derive from mesoderm like muscle, blood, and connective tissue precursors. Carcinomas are by far the most common type of malignant tumor, more than 90%. But tumors are solid masses, but the leukemias, a class of sarcomas, grow as individual cells in the blood. The name leukemia is derived from the Latin for white blood. The massive proliferation of leukemia cells can cause a patient's blood to appear milky. In this chapter, we first introduce the properties of tumor cells, illustrating how every aspect of cellular homeostasis and the interaction of cells with their environment is altered in cancer. We then discuss the origins of cancer and describe the evolutionary processes that lead to the formation of malignant, often metastatic cancers. Next, we consider the general types of genetic changes that lead to the unique characteristics of cancer cells and the interplay between somatic and inherited mutations. The following section examines in detail how mutations affecting both growth promoting and growth inhibiting processes can result in excess cell proliferation. We conclude the chapter with a discussion of the role of cell cycle deregulation in cancer and how the breakdown of genome maintenance functions contribute to tumorigenesis. First, how tumor cells differ from normal cells. Before examining the genetic basis of cancer in detail, let's consider the general properties of tumor cells that distinguish them from normal cells. The change from a normal cell to a cancer cell commonly involves multiple steps, such as one, adding properties that make cells more likely to grow into a tumor. The genetic changes that underlie oncogenesis alter several fundamental properties of cells, allowing those cells to evade normal growth controls and ultimately conferring the full cancer phenotype. Cancer cells acquire a drive to proliferate that does not require an external inducing signal. So, here we can see a picture which shows the overview of changes in cells that cause cancer. During carcinogenesis, 6 fundamental cellular properties are altered, as shown here in this tumor growing within normal tissue to other, and this give rise to complete most destructive cancer phenotype. Less dangerous tumors arise when only some of these changes occur. In this chapter, we examine the genetic changes that result in this altered cellular properties like resistant cell death, then sustained proliferative signaling, evade growth suppressors, then activate invasion and metastasis, enable replicative immortality, then induce angiogenesis. So that's what is happening here. Later, they fail to send signals that restrict cell division and they continue to live when they should die. They often change their attachment to surrounding cells or to the extracellular matrix, breaking loose to move away from their tissue of origin. Tumors are characteristically hypoxic or oxygen starved. So, to grow to more than a small size, tumors must obtain a blood supply. They often do so by inducing the growth of blood vessels into the tumor. In this section, we describe the characteristics of cancer cells. We first discussed the changes in the cancer cell's genetic makeup that affect virtually all cellular functions, allowing the cancer cell to escape proliferation regulation and acquire the ability to divide indefinitely. We then see how the genetic changes in a tumor cell and its interactions with its environment facilitate its escape from the constraints of the tissue it was once a part of and allow it to invade neighboring tissues and colonize distant sites in the body. Then, the genetic makeup of most cancer cells is dramatically altered. At the turn of the 20th century, David von Hansmann and Theodore Boveri first documented what we know what we now know to be an almost universal feature of cancer cells. Their entire genetic makeup differs dramatically from that of normal cells. Tumors harbor all types of genetic alterations, point mutations, small and large amplifications and deletions, translocations, and aberrant numbers of chromosomes, generally too much and too many, a condition known as aneuploidy. This recent sequencing of many human tumors have provided insights into the frequency with which these genetic changes occur in specific cancers and has identified new types of mutational mechanisms. Typical cancer cells exhibit whole chromosome or chromosome arm gains and losses involving a quarter of their genome. Local amplifications and deletions affect about 10% of the cancer cell genome. Perhaps the most surprising result revealed by the sequencing of cancers is the high degree of variation in mutation rates across different cancers. So, mutations are rare in pediatric cancers with substitution rates as low as zero point 0.1 base changes per megabase, but those rates may be as high as 100 base changes per megabase in mutagen induced cancers such as lung cancer and melanoma. DNA replication and chromosome segregation errors can lead to aneuploidy and to gains and losses of chromosome arms. Decreased replication fidelity and mutagens also profoundly affect the cancer genome. In addition to this well established genome instability inducing mechanisms, cancer genome sequencing has identified other novel, unusual methods and mechanisms that lead to the dramatic genomic changes observed in human cancers. Hypermutation, characterized by multiple base pair substitutions near translocation breakpoints, has been discovered. The mechanism underlying this dramatic local genome alterations termed catagesis. Catagesis. From the Greek for thunderstorm is not known, but probably involves an enzyme known as activation induced deaminase or AID that plays a key role in antibody diversity generation. And another highly unusual mutational mechanism that occurs in 2 to 3% of human cancers, but with a prevalence of up to 18% in aggregate in aggressive neuroblastomas, a type of nerve cell tumor, is called chromothripsis. Here, entire chromosomes or large parts thereof are shattered. Thriapsis in Greek means shattering and stitched together again in what appears to be a random manner, leading to dozens or sometimes even hundreds of rearrangements. This shattering and stitching together of individual chromosomes appear to occur when chromosomes of parts therefore are not incorporated into the nucleus but rather from their own micronuclei. This replication occurs less effectively leading to chromosome breakage and stitching together of chromosomes pieces in a random manner by non-homologous end joining, a form of DNA repair in which DNA pieces are fused together via their ends. As we will see in the following sections, the genetic changes that occur in cancer cells affect virtually all aspects of cellular homeostasis, proliferation, tissue organization, and migratory properties, as well as survival and proliferation at foreign sites in the body. Then, cellular housekeeping functions are fundamentally altered in cancer cells. Cancer cells can often be distinguished from normal cells by microscopic examination. They are usually less well differentiated than normal cells, and cancer cells frequently exhibit the characteristics of rapidly growing cells, a high nucleus to cytoplasm ratio, prominent nucleoli, an increased frequency of mitotic cells, and relatively little specialized structure. Tumor cells differ from normal cells not only in their appearance but in their entire protein composition. Genomoid gene expression and protein composition analysis have shown that in all organisms studied to date, increase in gene copy number lead to corresponding increase in gene expression. Notable exception to this observation are the gene located in sex chromosomes. Thus, the genomic changes so characteristic of cancer cells, losses and gains of whole chromosomes or chromosome parts have a profound effect on the protein composition of the cell and hence on most, if not all, cellular functions. This in turn causes a stress response that is aimed at offsetting the protein imbalance that cancer cells experience. For example, cancer cells rely heavily on protein folding and degradation mechanisms for their survival as a direct result of their dramatically altered chromosome composition. This dependence of cancer cells on proteome maintenance pathways for their survival makes the constituents of these pathways attractive drug targets. Another prominent feature of cancer cells is their use of an unusual energy generating mechanism. Normal differentiated cells rely on mitochondrial oxidative phosphorylation to satisfy their energy needs. Cells metabolize glucose to carbon dioxide by oxidation of pyruvate through the tricarboxylic acid cycle. Cycle in the mitochondria. Before learning that, let's see this picture of normal karyotype and cancer karyotype. So when we look at this picture, we could see cancers have highly abnormal karyotypes. Highly abnormal karyotypes. So image of chromosomes obtained from a normal human cell with its characteristic 23 pairs of chromosomes and B from a SW403 colorectal adenocarcinoma cell line. Each individual pair of chromosomes has a distinct color. Two characteristics of cancer cells are evident. First, the number of individual chromosomes are altered compared with normal cells. Second, many chromosomes are composites of pieces from different chromosomes. So that's the difference. Only under anaerobic conditions do normal cells undergo anaerobic glycolysis and produce large amounts of lactate. Most cancer cells, however, rely on glycolysis for energy production, irrespective of whether oxygen levels are high or low. The use of glycolysis to produce energy, even in the presence of oxygen, called anaerobic glycolysis, was first discovered in cancer cells by the biochemist Otto Warburg and is therefore called the Warburg effect. Warburg effect. The metabolism of glucose to lactate generate only 2 ATP molecules per molecules of glucose, while oxidative phosphorylation generates up to 36 molecules of ATP per molecule of glucose. When we look at this picture, differentiated tissue is there, then proliferative tissue will be formed and tumors tissue is there. Differentiated tissue, glucose formation, then pyruvate, then that lead to oxidative phosphorylation, but lactate is produced in another case, so anaerobic glycolysis will be there. In proliferative tissue or tumor, glucose, pyruvate, lactate are formed, or CO2 or anaerobic glycolysis or Warburg effect will be produced. So, energy production in cancer cells by aerobic glycolysis. In the presence of oxygen, non-proliferating or differentiated cells metabolize glucose into pyruvate via glycolysis. Pyruvate is then transported into mitochondria where it is fed into the TCA cycle. Oxygen is required as a final electron acceptor during oxidative phosphorylation. This, when oxygen is limiting, cells metabolize pyruvate into lactate, allowing glycolysis to continue by cycling NADH back to NAD+. Cancer cells and proliferative cells convert most glucose to lactate regardless of whether oxygen is present or not. The production of lactate in the presence of oxygen is called aerobic glycolysis. Cells can, however, use glycolysis intermediates to synthesize macromolecules and lipids. The rewiring of glucose metabolism could thus provide the fuel necessary to sustain macromolecule biosynthesis and proliferation of cancer cells. Not only do cancer cells rewrite their metabolic pathways, but some cancer types produce novel metabolites that play a critical role in disease. 70% of glioblastomas, oligodendrogliomas, and astrocytomas all are brain cancers, and approximately 25% of acute myeloid leukemias harbor mutations in isocitrate dehydrogenase or IDH, a TCA cycle enzyme that converts isocitrate to alpha ketoglutarate. The IDH mutations found in these cancers cause the enzyme to convert isocitrate into a new metabolite, 2-hydroxyglutarate, which accumulates to levels of up to 5 to 35% in cancer cells. So, how long 2-hydroxyglutarate promote tumorigenesis? It inhibits several enzymes that require alpha ketoglutarate for their function, including proteins that regulate the methylation state of histones. In this way, 2-hydroxyglutarate alters gene expression. Whether 2-hydroxyglutarate is the only example of a cancer-specific metabolite or whether it is the first in new class of oncometabolite remains to be seen. Then, uncontrolled proliferation is a universal trait of cancer. We know that. In normal tissues, cell proliferation is a tightly controlled process. Growth promoting factors are released in a highly controlled fashion to ensure that cells proliferate only to replenish a tissue. Cancer cells have evolved mechanisms to escape these tight controls. Cancer cells upregulate growth promoting pathways while simultaneously downregulating growth inhibiting and cell death pathways. In this manner, cancer cells gain the ability to proliferate continuously. This ability leads to the expansion of cancer cell population. As we will see, selective targeting of the mutations that cause uncontrolled proliferation is highly successful approach in treating certain types of cancers. Increase in proliferation promoting signals and decrease in proliferation inhibiting signals are not the only changes that endow cancer cells with the ability to proliferate indefinitely. Chromosome ends need to be protected throughout cancer cell proliferation. Telomeres. The physical ends of linear chromosomes consist of random arrays of short DNA sequences, TTA, GGG, invertebrates. That we'll be learning later. But before that, we could see some major picture like 2-hydroxyglutarate is a cancer specific metabolite. So isocitrate dehydrogenase IDH is an enzyme in the TCA cycle. It converts isocitrate into alpha ketoglutarate, which is then further converted into succinyl CoA. Some mutant forms of IDH found in many brain cancers create a novel metabolite like 2-hydroxyglutarate 2HG from alpha ketoglutarate. 2-hydroxyglutarate accumulates to extremely high levels in the cells and inhibits the activity of proteins that require alpha ketoglutarate for their function. Many of the enzymes that are inhibited control gene expression such as TET family of DNA hydroxylases are the methylation state of histones, such as histone methyltransferase and demethylase. So we could see acetyl CoA is there that will form citrate. This is aconitate and isocitrate. Then wild type isocitrate dehydrogenase, mutant isocitrate dehydrogenase found in cancer. That will lead to 2-hydroxyglutarate will be there. Then histone methyltransferase will not be functioning after that. Tet DNA dehydroxylase is also not functioning. Then histone demethylase got inhibited. So because of that, the normal pathway of TCA cycle is disrupted. So all the other products like succinyl CoA, succinate, fumarate, malate, oxaloacetate, etc. are not formed. Inhibition is happening there. Then telomerase, a reverse transcriptase that contains an RNA template, repeatedly adds TTA-GGG repeats to chromosome ends to lighten or maintain the 320 kb regions of repeats that decorate the ends of human chromosomes. Then embryonic cells, germline cells, and stem cells produce telomerase, but most human somatic cells produce only a small amount of telomerase as they enter S phase. As a result of other modest telomerase activity, their telomeres shorten with each cell cycle. Extensive shortening of telomere is recognized by the cell as a double strand break and consequently triggers cell cycle arrest and apoptosis. Then tumor cells overcome this fate by producing telomerase. Many researchers believe the telomerase expression is essential for a tumor cell to become immortal. Indeed, the introduction of telomerase producing transgene into cultured human cells that otherwise lack the enzyme can extend their lifespan by more than 20 doublings while maintaining telomere length. The reliance of many cancers on increased telomerase activity has led some researchers to propose that inhibitors of telomere could be highly effective cancer therapeutic agents. Next topic, cancer cells escape the confines of tissues will be covered with the help of next topic. Thank you for listening. I'm sorry, next video. That topic will be explained with the help of next video.